Welcome to the video tutorial about how to analyze the data from an ELISA test using the Discngine Assay platform.
Let's start by introducing the solution. Discngine Assay is a cloud-based platform that handles all your plate-based data related to screening campaigns from plate preparation to the results analysis process using advanced analytics. The goal of this study is to identify compounds that can inhibit HIV-1 replication in latently infected cells. For the ELISA test, a microplate has been prepared using different concentrations of HIV P24 antigen as standards, some controls, and the samples using two types of clones of infected HIV cells incubated with the different compounds we want to test. As a result, the optical densities of the microplate have been measured at two different wavelengths. Using Discngine Assay, we will generate standards curves using the different concentrations of HIV-1 P24 antigen and the measured optical densities for each concentration. Then optical density values of the samples will be interpolated in the standards curves to quantify the concentration of P24 antigen using linear regression. Depending on the concentration of P24 antigen measured in the samples, we will be able to identify which samples present the best inhibition performance and can be selected as hits. Let's start with the analysis. We are now on the homepage of the Assay application. The first view displays the history of the analyses performed. Let's create a new run. We want to create the new assay from a template, so we will select the execution mode. We will be guided by the workflow template during all the assay. We will use the template named HIV-1 ELISA test, created previously in design mode, and we will name it. Let's go now to the file reading step. The parameters like the plate format and the reader selection are defined automatically by the template. We will just upload the file containing the raw data results provided by the reader. And we will go to the next step. The system has automatically filled the mode, the number of measurements, and the number of plates defined by the template. Also, the readout values have been matched with the optical densities measured at two wavelengths, and the ratio of both measurements has been calculated. Let's go now to the plate mapping step. Controls and doses layouts are mapped automatically by the system. We can also load barcode information, or go to the well validation step. For well validation, predefined data normalizations have been calculated from the raw data file as the average subtraction to remove the optical density signal of the blank. If we take a look to the visualizations, we can see the plate layouts and the optical densities obtained for the controls and the standard. The z-score and the plates factor calculated are displayed in this visualization. In the next window, we can have a look at the calculated concentrations for all the plates. We have also the data tables with all the numeric data from data normalization calculations. In order to proceed with the quality control process, we will invalidate some anomalous points following different business rules. For example, we can invalidate the anomalous value of the positive control signal and the system will recompute the results. We can do the same for the plate z-factor, for example, invalidating points that have a z-score factor lower than 0.5. The results will be recomputed. Now let's take a look at our reference curve, so we go on reference analysis. Here we can see the standard curves generated for all the plates. These are the curves that we will use to calculate the concentration of P24 antigen on the samples. We can also invalidate some points if we consider it necessary. Once we have finished our QC validation step, we can go to the sample mapping step. Here we will edit the sample information. To do this, we will use a file again that contains information about samples, doses, and experimental conditions of the assay. Let's upload our file in the system. Once uploaded the file on the system, all the information will be mapped for all the plates. We can see plate-by-plate information like the sample number, the concentration, and also the experimental conditions as the type of cell that has been used in the sample preparation. If we continue with the workflow, an overview of the sample mapping will be displayed. We can edit if needed, or go to the sample analysis step. Here we can visualize the results of the sample analysis. The first visualizations show us which compounds demonstrate the best inhibition performance of P24 productions for both cell lines with a concentration of antigen measured lower than 8 nanogram per milliliter. Below we can see the compounds that could be retested as concentration of P24 is between 8 and 20 nanograms per milliliter. And then we see the compounds that have no effect on the P24 production due to the high concentration of antigen measured in cells. Now let's select the samples that could be considered as hits and add them to the hit selection menu. We can export this selection by downloading or create a list in Sample application. Sample is the compound management module of Assay, so we can use this list in Sample to prepare a new plate and run further analysis. We have shown how to perform an assay with the execution mode. You can also design your assays in mode design, save them as templates and execute them with execution mode as we did in the video, or even use them in the autopilot mode, where the assay will be run automatically.
Thank you for listening, and if you want to know more about our Assay platform, do not hesitate to contact us.